we didn't go that far. But someplace I'd like to go, there was a supplemental challenge that was to predict diseases. You know, these 145 people said we have these diseases.
Exactly. That's exactly what this was.
But you could...
for the contest, we had the training data, and the training data enabled us to build a model. And then for the contest, we were supposed to take the model that we built and then go on and try it for the 23 people, which were the blind situation, and see if we could predict for the 23 people. Yes. So we did. That's still in the future.
But beyond that, you could predict diseases that nobody reported if you take everything in GWAS and match them against all the genomes and say, well, we think this person could have this other disease that they don't even know about.
Well, if you have the statistical probability from GWAS that's correct, will you be able to do a statistical hypothesis test comparing it to your data set with...
That I'll let David speak to that. Yeah,
okay.
Good point. See, there we go.
Oh no, I'm just playing. I think the answer is yes. The three of us aren't that great at statistics, so we didn't do any. We didn't do that.
We didn't use a statistician. Or if we have any volunteers, we really could use a statistician.
You're hinting at machine learning too.
And yes.
And there's libraries that might get you halfway there without being a statistician.
Okay. Let me just jump in on that one, because I think that, you know, that's a perfect lead-in. Okay. What ended up happening, and David, correct me if I'm wrong, but of the people that actually completed the challenge, one group used one of those machine learning type models. Yeah, a heavy statistical model, and they were the ones that won the challenge.
Well, is there a known solution to the challenge that's the best?
Yes.
So it is solvable.
I'll let David came up with this.
I didn't see that. I don't think there's a...
There were...
Well, they send you the answers. We have the answers.
We have the answers, so we could back test.
But there really isn't a solution. That's why they're doing this challenge, so we can come up with solutions.
There's the answer, those 23 people, like self-reporting their diseases also, like the training data that you got, right? It was all self-reported diseases,
Right.
and SNPs that you were trying to correlate.
Right.
So with the 23 that was the test set, it was also self-reported data that they were, and that's the answers.
And it was 145 that was a test set, right? Do I have that right? 145 was the test, the training set, where it was like from previous years, and we could use that to train because we had all the answers. But then they would give, in order to complete the challenge, you had to take 23 where it didn't give you the links,
Right.
and then you had to
It's just the self-reporting thing that makes it seem a little tricky to me. Because like, so then you get the answers, right? But it's only what those 23 people self-reported.
Right.
What happens, but those 23, the standard of error that you'd experience in those 23 is the same as the standard of error that you're going to have in the other set.
Which is true. But I sort of wonder, an interesting follow-up to this is like, if your model predicts some of these diseases that these 23 people might have but didn't self-report, it'd be interesting to go see if they actually have those. Then your model would be even better than the answers.
That was one of my nightmares, actually. That was one of my nightmares that we would get, and we would say, okay, you're going to develop, you know, astigmatism or something serious, ulcerative colitis or something, that our mathematical model says, that predicts that you're going to develop this. And that was one of my nightmares. What do you do in that case? Yeah.
So the data you guys got was the full genome of each of the 145 people, and then you guys also got data with, did you guys have to parse that to get SNPs out?
Okay, this is a section of it. It was a two-step process. The data for the full genome was run through a program which extracted the SNPs and came up with a text file called the VCF file that contained the 4 million SNPs and 10 million lines of text. So, you know, it was a two-step process. We got the VCF files. We didn't get the original genomic data, the 3 billion letters, no.
Yeah, we have the full genome.
Do we have those too?
It was the same file, and then we extracted the SNPs from it.
Okay.
So, yeah, the VCF is pretty close to raw, but it is a little bit worked up. It's not like truly, you know, whatever,
They're coming up with 2 new
already
ones, did you
there.
hear that?
But,
No, really.
but yeah, the VCF files are a little bit less raw than really raw, but they're pretty raw, and there's 10 million lines in there. But then,
Really?
yes, to answer someone's question here, we did have to parse quite a bit just to get the pieces of information out that we wanted to care about, because on each of those lines, there's 20 other fields of data that at this stage and with our kind of just being very new to this, we didn't want to take a look at any of that other stuff that we didn't really know what to do with it or if it was relevant to us. So we really focused on the RSIDs and just a few other, maybe a few other fields.
We didn't look at things like structural variations or insertions or deletions, but still there would be a lot rows like that's one row from, I don't know, I can't really point, but we parsed the 2 SNPs out of that one nucleotide had 2 SNPs associated with it. But other rows in a BCF file would have 30 or 100 nucleotides all in the same row, and I didn't do the research to find out what those are, why they look like that, but because we only wanted to get the SNPs.
So it seems like every SNP is really 3 SNPs, because if the canonical or reference one is an A, it could be a G, T, or C.
It can be, yeah.
So I wonder if it would be interesting or help statistically to kind of break it out into the 3 SNPs and see if that changes the data at all.
Totally agree, but as I understand it, GWAS didn't break it out.
Right.
But we did make sure, oh yeah, GWAS did break it
Does
out.
it break it
And we
out by
made sure
nucleotide?
that
Okay.
the SNP, that the person's allele matched GWAS's allele.
Okay.
But not every allele has a different RSNP, is that?
I think that's probably true, but we made sure they matched.
Yeah, that was actually one of the things that I think that bears further research, because especially for those RSIDs that are one allele is protective and one allele is just the opposite, like in the case of there's a lot of cases where you just get that one shift and it's protective or not. I think that it bears looking into that a little bit further to verify that they haven't actually said, okay, for this one it's benign, this substitution is benign. That is a totally different RSET than this one that is protective, or this one that's not benign, that's harmful. So I think that's something that I have been confused about, but sometimes that wasn't even in the data though, so it's hard to tease that out.
It would be easier to predict disease sets rather than single diseases, where
Like,
That's a very good
yeah, let's
question.
do the study.
And that gets back to the idea of multivariate statistics. David, could you back up to your future direction slide, please?
Not many words on it.
Keep going.
That one?
Keep going. It's the one that talks about finishing the challenge and using multivariate statistics.
Oh, yeah, that's in the notes now.
Oh, okay. Could you go back to that one?
Too many words. If we get to the notes format, I don't know how to do that.
Oh, okay.
You have to go to the laptop?
But, one of the things that, pretty much came out of the study is there's an interrelationship. It's not a one-to-one. And, maybe there are families that go together, that if you have one particular set of conditions, that it's a family that comes through. And certainly this is a multivariate statistic. And I was hoping for the slide to show you, but, it's the kind of thing that was one of the things we talked about, going into it with a machine learning. And we did originally modeling with neural networks for exactly that approach, okay? But we didn't get the results we were looking for, so.
Probably because of the bug I had.
Yeah.
You could try again.
One question. How did you choose in the beginning to choose asthma and acne? What made you choose
Oh,
those?
we just went in alphabetical order. Almost.
Remember the slide that had all the boxes?
Yeah.
There were 2 that had lots and lots of people. so that we had a large statistical sample,
Yeah.
and that's why.
So you showed, so asthma, just like you showed on the slide, is really a name of like you were talking, a set of diseases, a set of conditions, if you will, that have those phenotypic expressions. So genetically, they're probably so varied that
I totally agree.
145 people is not gonna.
I totally agree.
So there were, I think there were diseases on there that are much more narrow and much more defined than you could have.
And that's one of the things we learned as we got deep into this. You were totally correct. You know, that might not have been the one for us to focus in on.
Yep.
Well, I just wonder for future directions, if you just took 3 of the less common ones that are more defined diseases, and did the exact same process, if you have, more reliability, in your data
Oh.
that comes out.
Well, it's all in there. We just presented those two, but we have results for everything.
We have results for.
Did you ever look at, like, the ones that are less reported, that are more specific? Like, there's certain diseases like,
Yeah,
Crohn's
Graves' disease is extremely narrow.
Yeah.
So if you guys look at that.
That's why it
For
scored,
Crohn's.
it scored second high of everything we had, I think. But that's partly
Oh, so
because.
those are your disease, those are your disease results, like your beta scores?
Yeah, the size of the circles is proportional to basically the strength of the linkage. So,
Oh, okay. Can we see that other one?
so Crohn's was in 6th place.
Yeah, I see. That makes sense, please.
But that's when we added the beta, so I don't totally trust these results.
Yes.
And this is also dependent on how many people reported it. I think only one person might have reported Crohn's.
Yeah.
What did you guys find in terms of people out there on the internet doing this sort of stuff and willing to engage you with questions and expertise and things like that?
No. From my memory, okay, we did not go onto the internet and, look for more information, except from the PGP people, and David engaged with them.
It's mostly universities. We were one of the first non-universities to join this competition. There was one group in San Francisco that did also.
I did a little bit of looking, like I said, can we find at least some kind of blueprint here? And there were some papers that people had, talked vaguely about their process, or that, they applied a statistical model or they did machine learning, but no one got into the nitty-gritty of, like, for instance, how do you parse these things? How do you, what do you throw out? What do you take as far as good data sources and bad data sources? So, I don't know if that's because everyone's competitive, or probably just because there's not a whole lot of people doing it out there and publishing their work. I think we haven't published any of our information. We're just hacks, but we had a good time.
We learned a lot.
Yeah.
And we have all the published results of, there's some things we're not supposed to share. I think you had to be a member, but if you want to see them, I'll show you. One-on-one.
So what are you guys gonna do next? Or are you gonna continue this, maybe finish the challenge, or, 'cause you have my 23andMe data still, are you gonna do anything with that?
Yeah, we sort of have a pipeline, but I'm a pretty sloppy Python programmer, so maybe I would like to have a team that cleans it up and maybe use GitHub or something to work on it together. But it wouldn't be hard using Python to just stick 23andMe in instead of these genomes. But I have a feeling, because there isn't going to be much there, because there's only 600,000 SNPs. But I want to do it because I'm narcissistic.
I'm like Mark, though. I don't know if I want to know that I have Huntington's disease or those things.
I don't want to know.
Part of me at least doesn't want to know. I probably should stick my own genome. I think that's a good idea.
I'll just add real quick, if any of you guys are interested in joining and working on this, we recently got organization status with GitHub, and Dylan actually is administrator of it, and so
As
Deborah Biles,
So
yeah, so as an organization.
we set up a repository for
Yeah.
this